Good afternoon, students. So here's part B of lecture number 17 on translation. In this particular lecture, we're going to be talking about mutations and how those work. So firstly, let's just talk about what a mutation is. There's many different types. Most mutations we'll talk about in this class are going to be called point mutations. So what does point mutation mean? It means there's a mutation at a single nucleotide. So take a look at this particular codon here. This is a codon for a gene in the beta globulin gene, and this is part of our hemoglobin. And if you take a look here, we have a codon. So you can see we have GAG is the codon. The complementary strand is CTC. The messenger RNA here, of course, is GAG as well. If you look that up on your genetic code, we get a glue. So glue as glutamic acid. So that's the amino acid. So this is the normal wild type version of this particular gene for normal hemoglobin. We're going to take a quick look at what happens if somebody has something called sickle cell anemia. So in sickle cell anemia, their blood cells, notice normal red blood cells are nice and round, and sickled cells look like sickles. So what's a sickle? It's this kind of thing. You may see in cartoons. The Grim Reaper has one. Kind of looks like this. There we go. That's a sickle. Anemia means you don't have enough blood. So we'll talk about what that means in a minute. Let's take a quick look at the mutation though. So notice we have a single mutation here. This A is here. Before we had a T, and now it's changed to an A. We're looking at the top strand. The bottom strand we had an A that is changed to a T. So one small mutation. So now our codon, this GTG, becomes a GUG on the messenger RNA, and the final amino acid is a valine. So you may not know your amino acids very well, but this, I said, is a glutamic acid. This is acidic. It means it has a charge on it. Valine does not. This is a non-polar amino acid. It is hydrophobic. So we're changing the chemical properties of this amino acid, this one spot on the protein. So how does that work? How does this go from a nice round cell to a sickle cell? Let's take a look. So what happens here is that particular amino acid happens to be on the surface. So they're showing it right here. I'll just show you, there's an arrow. That little amino acid is shown there in green. It happens to be on the surface. So something that is a glutamic acid is actually polar and it's water soluble. Now what I have is a valine. A valine is non-polar, hydrophobic, and does not want to interact with water. So what happens is all these hemoglobins, so this is one hemoglobin here, this is another hemoglobin here, here's a third one. They're normally soluble, and now instead they're clumping together. It's kind of like having a little sticky patch, that hydrophobic component. And they can stick together, and rather than having nice soluble proteins, you end up with these filamentous structures inside your red blood cell. So here's what the red blood cells end up looking like. Rather than being these nice round things, these are relatively flexible, can fit through the capillaries. These sickled cells are a little bit more rigid and they break easily. So this is why people with sickle cell anemia end up with anemia. Lots of the red blood cells break open. Apparently it can be quite painful in the extremities, along with a number of other symptoms. So sickle cell anemia is one of the first genetically inherited diseases that we as humans have understood to any level, and we actually understand it quite well. Take a look here on the left. This shows a map of people whose ancestors originated from these parts of the world. So you can see sub-Saharan Africa, a few parts in Europe, and a few parts in Asia and the Middle East. This is where the alleles for sickle cell anemia originated from. And here's something really interesting that we've found about sickle cell anemia, is that if you take a look at this second map, it roughly overlaps with the historical distribution of the disease malaria. So malaria is a disease that's spread by a mosquito. A parasite is spread by a mosquito, and it gets into the blood. So it turns out that if you have one copy of the sickle cell gene, you're probably going to live a normal life. If you have two copies, you're going to have sickle cell anemia. But there is an advantage of being a carrier to having one copy of the sickle cell gene, is that it seems to give people a much better survival rate when they get exposed to malaria. So this is one reason why this gene has probably stayed within the human population gene pool, and this is why it is featured a lot in genetics textbooks, because it's a very, very interesting case of a human genetic disease. So I want to talk a little bit about some other types of mutations and give them names in terms of what the effects are and some technical terms. And you'll need to know these particular technical terms for the final exam. So we're going to talk about no effect or silent mutation, missense and nonsense mutations. I'm also going to talk a little bit about these things called insertion or deletion frame shift mutations. This is what we were going to cover actually in Lab 10 with that mutation exercise that we never did complete. So first of all, let's talk about a silent mutation. So here's a little stretch of DNA. You can see there's the DNA right here. And what they've done is they've taken it and they've done the transcription. So our ATG is there. There's the start codon. Now we have AUG, AAG, UUU, GGC, and UAA. They've translated it, and we have methionine, lysine, phenylalanine, and glycine. So what happens in a silent mutation? Let's take a look. Silent mutation. Notice right here we have an A instead of a G. So that changes this particular C to a T, and it becomes a U down here. Take a look on your genetic code, and you can see if we go from a GGC to a GGU, we get glycine. So we went from glycine to glycine. So no change. This is why this mutation is called a silent mutation. We have a different base pair, but two different codons can code for the same amino acid. So in this case, we have a silent mutation. Lots of silent mutations are found in the human genome because, of course, there's no effect on the biology because we have the exact same amino acid, and therefore biologically there will be no effect on the phenotype. No effect on the amino acid sequence. Okay, let's talk about missense mutations. So missense, by the way, is the first half of the word mistake. So this implies we are going to have the wrong amino acid, just like sickle cell anemia. So let's take a look. Here's the example. You can see it's the same sequence given on the previous slide. They've made the messenger RNA and they've made the amino acid sequence. So let's take a look at the change here. There we go. In this case here, we have a T instead of a C. So this particular codon becomes, here's the codon here, AGC becomes, it used to be a GGC, and now it becomes an AGC. There we go. And now we're going from a glycine, and we are getting a serine. So now we have a new amino acid, a missense mutation. So in this case here, one amino acid is changed. In the case of sickle cell anemia, it can be quite serious if you go from an amino acid of one property to an amino acid of another property. If you go from polar to non-polar or the other way around, in some cases the change can actually be mild or not even noticed. If you change one polar amino acid for another polar amino acid, you could imagine the chemical effects are going to be a lot smaller. There's a lot of variables in here. You can learn about this more if you take more genetics and biochemistry classes, or send me an email if you have any questions. This is really, really amazing stuff. Okay, the last type is nonsense. So notice nonsense, the first part of nonsense is no, and in this case we mean we're going to have no amino acid chain. So let's take a look at that. Here's our wild type again with our DNA strand, our messenger RNA, and our protein strand. And let's take a look. So in this case here, we're going to change an A to a T. So this here is in the second codon right here. Initially, way up top, take a look, we had an AAG, and now we have a TAG, which becomes a UAG. So take a look at your genetic code. UAG stands for stop codon. So we are no longer going to get, there's going to be no lysine, no phenylalanine, no glycine, or whatever else is found on that protein. So this protein is going to be truncated. It's going to be cut short. So what does this mean for the protein? Well, this is very bad. The protein, if it's right at the end of the protein, you're only missing 2 or 3 amino acids, it may not be a big deal, but most of these mutations are going to be very serious, and the protein is going to be non-functional because it's missing some very essential amino acids. There's one genetically inherited disease that we know quite a bit about that is due to a nonsense mutation. This is a type of muscular dystrophy. And you can see it says here, this is a severe recessive X-linked mutation. That means it's on the X chromosome, so it usually only affects boys because they're more likely to get the mutation as opposed to girls that would have to have 2 copies of the non-functional gene. So this is basically leads to an issue in the person's muscular development, and they have all sorts of issues walking, and their bodies are not developed properly. Okay, so we were just talking about base pair mutations. I want to talk a little bit about insertions and deletions. You can see there are kind of 3 scenarios that may change what happens to the protein. So let's take a look at these. So the first frameshift mutation, you can see we're talking about immediate nonsense. So remember, nonsense means we're introducing a stop codon. So let's take a look at this example. So notice here we're putting an extra A in here right after the first codon. So we have a whole extra nucleotide, and now we end up, you can see here we had an AAG, so AAG up there. Now the U is added in and we get a UAA. So that's a stop codon. So now we have a truncated protein, and just like what we showed before, this is very bad for the protein and will probably lead to a non-functional protein. So second example here, we have extensive missense. So that just means we have the wrong amino acids. So let's take a look at this. Here's an example here. We've cut out an A. So a frameshift means you can insert or cut out a nucleotide. So cut out the A, and notice our consequence, we have a UUG. So instead of a phenylalanine up here, we have a leucine down here. And the next amino acid, GCU, becomes an alanine, and so on. So every single amino acid after this point will probably be the wrong one. This is also bad for most proteins because you have all the wrong amino acids, so you're not going to have the right protein. It's going to be totally non-functional. So here's another example. No frameshift. It says 1 amino acid missing. So what does this mean? Let's take a look. So in this case here, they are cutting out an entire 3 nucleotides. So of course that means 1 entire codon. So in this case here we have an AAG is missing. So we cut out our lysine, that's gone. And then we're just moving on to the phenylalanine and the glycine. So this, again, it may or may not be a problem. It really just depends on where in the protein and whether a crucial amino acid is missing or not. So cystic fibrosis is a good example of a frameshift mutation where an entire codon is missing, so an entire amino acid, and it turns out it's a quite crucial one. This is a recessive genetic disorder, and this is actually most commonly found in people of European descent or Caucasians. So about 1 in 700 of us are carriers. So if you're a carrier, you're not usually having any known phenotype, but if you have 2 copies of the gene, you end up with cystic fibrosis. So what is cystic fibrosis? You can see here are the details. We have a codon is missing, and this removes a crucial phenylalanine residue. And this particular gene is involved in a chloride secretion in the tissues. So this leads to all sorts of issues in the digestive system and in the lungs where you have secretion problems going on. We can correct some of them for people by giving them enzymes for their digestion. In the lungs, they end up with a really thick mucus, which can be a problem if they get an infection. Unfortunately, for people with cystic fibrosis, the average lifespan is somewhere in the low 30s, because often they will eventually get some sort of chest infection that will finish them off. So I wanted to finish off this lecture with one of these transcription questions with some mutations put in. So remember, this will be part of your final exam. You'll have to know how to do this for your final exam. So here's a question. As you can see, it's asking for 4 parts, and I have some different mutations that are going to happen. But before we get there, we need to do our transcription and translation to see what is going on. And so this would be a good review, so why don't we start there? So let's do the transcription and translation. So remember, the very first thing I want you to do is find that start codon. Do you see it? There it is. So remember, we are not looking here. This is not the start codon. Remember, that says 3' to 5'. Our start codon is this one here in the 5' to 3' direction. So that's my start codon. Next thing we're going to do, we're going to make our messenger RNA. So the messenger RNA, we're going to copy out this strand here. So this is the sense strand. We're going to copy it out, we're going to change all our T's to U's. So let's do that. Here goes. So there's our messenger RNA. Next thing we're going to do is we're going to find all our codons. So I'm going to underline them all triplet at a time, starting with the start codon. Here goes. So now that I have all the codons sorted out, we can start here at the start codon and we can do our translation. So our protein sequence, I'm going to use the genetic code that's found up there in the corner, and we're going to start off. The first one is, of course, methionine. Hopefully you have that one memorized by now. UAU is tyrosine, CCU is proline, and UAA, remember what that one is. Take a look up on the chart. This is the stop codon. So I don't need to write stop. I can just finish there. So my amino acid sequence is going to be methionine, tyrosine, proline. Okay, so let's try to answer those questions. I'm going to squish this up a little bit and squish that genetic code so that we can see everything on one screen for each question. So here is the first question. It says, what would happen if the T at position 1 was changed to a C? So let's take a look. We have our transcription and translation done just as before. Now we're going to take a look particular here at the second codon. So this TAT. So what's going to happen? We're going to end with some new DNA. The new DNA is not going to be TAT. The question says it's going to be TAC. So once we have our new DNA, we can determine what our new RNA is going to be. So that's going to be a UAC instead of a TAC. Remember, all our T's get changed to U's. And then we're going to look up our new amino acid. So UAC on the genetic code is tyrosine. So we're going from a tyrosine to a tyrosine. What is that? That is, of course, a silent mutation. So that would be the answer to that question. You need to give the full answer and say that we have a new amino acid or an unchanged amino acid and what it's going to be, and also give the correct terminology. Say that this is a silent mutation. Let's take a look at part B. Part B says, if the G at position 2 was changed to an A. So start off with our transcription and translation so that we can see what is going on. So now we are looking at, it looks like we're looking at the third codon in this particular sequence. And this particular G, notice it is on the template strand, not the sense strand. So this is going to make the whole thing just a little extra confusing, just a little extra step to sort things out. So let's take a look. What is our new DNA going to look like? Well, I'm looking at that bottom strand. The question says change the G to an A. So I'm changing this bottom strand. Instead of a GGA, instead of a GGA, it's a GAA. That means the top strand is now going to be a CTT instead of a CCT. Next, I need to figure out what the new RNA is going to be. So the CTT is now a CUU in the messenger RNA. I can take a look up on my genetic code and see what that is going to change. And our new amino acid is going to be a leucine. So I'm going from a proline to a leucine. Okay, so which one is that? That is a missense mutation. So remember to use your correct terminology. This is a missense mutation. Okay, part C. What if the T at position 1 was changed to a G? Okay, so there's my messenger RNA and my amino acid sequence. I'm looking at codon number 2. And what does the question say again? What if the T is changed to a G? So my new DNA, I'm going from a TAT to a TAG. Got to look up the RNA and see what a TAG, convert the DNA to RNA, which is going to be a UAG. Look up on my genetic code, what's a UAG? A UAG is a stop codon. So this is going to be a nonsense mutation. So there's an example of each. We had a silent mutation, a missense mutation, and a nonsense mutation. Okay, one more question, part D. It says, what if the C at position 3 was changed to a T? Okay, so there's our messenger RNA and there's our amino acid sequence. So take a look. We have our ATG. So here we go. There's the first codon. Here's our second codon. Okay, here's our third codon. So we've got methionine, tyrosine, proline, and then over here we have our stop codon. So it turns out that this particular codon, it's not even part of the gene sequence. It's actually after the gene. So what does this mean? It means it's not going to affect the amino acid sequence. So does this have a name? No, not necessarily. It doesn't have a special name. It just means the mutation does not affect the sequence. So you may have to write something like that, an explanation of what is going on. There is a caveat. It says there that this may affect some sort of regulatory component of the gene. Remember, we have a terminatory region at the end of the gene, and this may affect some sort of regulation of the gene. But that's kind of beyond this course, so we're not really going to talk about that any further. Okay, so just before we finish up this particular lecture, I just wanted to remind you that we're going to have a question on the final exam addressing all the types of RNA. I told you there were 6 types. This slide has 7 types. Notice I'm distinguishing between pre-RNA, pre-messenger RNA, that's before it gets processed, capped, and spliced. And then number 1 is messenger RNA, 2 is ribosomal RNA, 3 is transfer RNA, 4 is telomerase RNA, 5 is small nuclear RNA, and 6, we haven't talked about 6. This is signal recognition particle RNA. We are going to talk about that in the next unit. So stay tuned for that. Topic 17, summary, all on translation and mutations. Make sure you take a look at some of these internet tools and resources. I will put them on the course playlist for you to check out. Some of them are better than others, but hey, they're pretty good ways to review what we've learned. Don't forget to take good notes and take a look at those transcription translation problems. Here are some concepts. You can read them on your own time, download the PowerPoint, or they're in the notes. Don't forget to take a look at these study questions. Usually I have them as relatively big, open-ended type of questions, because usually when I make my final exam, I do look at these study questions as inspiration for my exam questions. I hope everyone is having a great day. Stay healthy. Let me know if you have any questions. Take care.